physically due to unforeseen circumstances. Anyhow, I hope you can still enjoy my talk online. Should you have any question, I would be happy to answer them later on. Okay, so here are the things that we are going to cover in this talk. First, I'm going to briefly discuss what functional MRI is about. I believe most of you are familiar with fMRI. However, some of us may be new to this field and would like to learn a little bit more about it. Next, I will talk about the main types of experimental paradigm design used commonly in many types of task-based fMRI studies. This includes the block design, event-related design, mixed design, and sparse temporal sampling design. And I'm going to finish off by providing a summary of these different designs. Okay, let's first look at the differences between structural and functional MRI. The figure on the left is an image of structural MRI, whereas the figure on the right is an image of functional MRI. So what is the difference between structural and functional MRI? In structural MRI, we can only see the anatomical structure, like in this case, the brain. However, from looking only at the anatomical structure image, we could not assess or determine what are the function of the different part of the brain. Here is when functional MRI comes to play. If we look at the functional images, here, the brain areas with different shades of grey are parts of the brain which are activated when a person is performing a task or given a stimulus. These are actually images of my brain when I was doing a pilot study for my PhD. So here you are looking at the image of my brain. Okay, these are also images of my brain after post-processing. The red and yellow areas are the brain areas which were significantly activated when I was listening and memorized a list of words. If you have good understanding of neuroanatomy, you will see that these areas are the auditory cortex, which is responsible for our hearing, and the prefrontal cortex, which is responsible for high-order cognitive function such as memory. Okay, let's do a quick task-based fMRI experiment. So here, you are looking at an image of a lady. From your point of view, do you think this is an image of a young or an old lady? I'm going to give you 5 seconds to think about that. Alright, time is up. So I believe you have your answer. Keep your answer to yourself. Okay, some of you may see the image of a young lady, while some of you may see it as an old lady. Whatever it is, I hope none of you see an image of a guy, unless you are looking at me here. Okay, well, according to this study here, it was found that participants' responses were based on their age. For example, participants who were young would see the image of a young lady, whereas participants who were slightly older say that they see an image of an old lady. I do not know whether this finding is true for you. However, I did try this test with my students in my class, and a majority of them who are young see an image of a young lady. So I see the image earlier as a young lady, possibly because I'm still young. I don't know. Okay, moving on. Okay, so let's look at what actually happened in our brains when we were doing the task earlier. So in a task-based fMRI study, the image that we saw earlier is considered a stimulus. Okay, so this image we call it stimulus. When we were looking at the image, the neurons activity in the part of the brain that controls our vision is increased. As such, more oxygenated blood will flow to that area of the brain in order to fulfill the high cognitive demand. The increased flow of oxygenated blood or bone signal can be measured using fMRI. However, it is important to note that there is a slight delay between the delivery of stimulus and increased blood flow. The delay is known as time to peak. So time to peak is the duration it takes for the bone signal to be at the highest following stimulus presentation. So in this case, we can see that the best time to scan the brain is 6 seconds after the stimulus presentation, because this is the duration that it takes for the bone to reach its highest level after stimulus presentation. If we scan too early, let's say we scan here, okay, or too late over here, we will not get a robust and reliable fMRI signal. So the best time to scan is between 5 and 6 seconds. So this is when the best signal that we can obtain. It is also important to keep in mind that the time to peak may differ according to the type of stimulus. For example, the time to peak is longer for an auditory stimulus than for a visual stimulus. So in this case, is an example of visual stimulus. So let's say that you are hearing a music or you are given a speech signal. The time to peak may be a little bit longer, okay? Okay, so here we have learned that paradigms refer to the strategy used to activate the brain. So before we conduct any task-based fMRI studies, it is important for us to make sure that the stimuli are presented accurately. The stimuli are presented and measured accurately. If not, the signal that we acquired will not be reliable to generate valid results. Therefore, we need to first know what we want to look at. From here, we then design a suitable experimental design. To do this, it is important for us to at least have a superficial understanding of the different types of experimental paradigm design. In the next slide, I will show you the types of experimental design used commonly in task-based fMRI studies. Okay, the first one is the block design. It is the earliest and simplest design and used in many task-based fMRI studies. In this design, stimuli are presented consecutively within a series of blocks. Let's look at the figure here, over here, okay? So we can see that the stimuli from one condition are presented in a block, followed by a stimuli from another condition. This design is also known as AB block design. Let's say you have several designs of baju raya, and your friend also designed her own version of baju raya. So you want to compare which design of baju raya will increase activation in brain areas associated with the reward pathway. So one way you can identify this is that in condition A here, you can show participants or your potential buyer 5 images of the baju raya which you designed. And in condition B here, you can show 5 images of the baju raya which your friend designed. By doing this, you can compare the brain activity of the participants when they view your design of baju raya and your friend's design of baju raya, and see which design produce higher activity in the reward pathway. Of course, you would hypothesize that higher activity in the reward pathway is associated with higher chances of purchasing. This is just an example of what we can do using this experimental design. Here, I'm showing you the block design that I used in my PhD project. You can see that trial 1 is actually condition A. Okay, from 0 second to 12 second, trial 1 is one condition, where I measure the participants' brain activity at rest, meaning they were not doing any task. And in trial 2 here, it can be considered as condition B, where I it's the active condition. It is where I measure the participants' brain activity while they were performing a memory task. Each condition is interleaved with one another. What I mean here is that there is condition A, condition B, and then repeat condition A and condition B. So this is example of block design. Here is another example of a study using block design, where the researchers were interested to see how the brain responds to food images as compared to non-food images. So what they did was that they showed several food images to the participants in the food block or condition A. So in this block here, the researchers show images of food, for example, cake or hamburger, to the participants. After a certain period of rest, between 8 to 12 seconds, the researchers then presented several images that were unrelated to food. For example, here we can look at the highlighter markers and pencils in the neutral block or condition B. So what the researchers were actually doing was that they were comparing the brain activity when participants looked at the food images and when they were looking at non-food images. There are a few things that we may want to consider when using block design. The first thing is the duration of the block. There is no specific duration. However, it depends on the type of the stimulus we are using. To identify the appropriate duration, it is best to look back at the current literature related to our study. However, most task-based fMRI studies use a block of approximately 15 to 30 seconds, with 15 seconds having been suggested to be the optimal duration. Another thing we should consider is the order of conditions. It is recommended for researchers to counterbalance conditions across subjects of the same study. What is meant here is that if we have two groups of participants, the number of condition A and condition B should somehow be similar in both groups. Okay, the advantages of block design are that it is a straightforward approach because we give a task or stimulus to the participants and we immediately measure the evoked brain activity. This design is known to have good statistical power, signal amplitude and robustness. This means that the signal acquired reflects the neural mechanism underlying a task or given stimulus. Therefore, this design is highly recommended for researchers who are new to functional MRI. However, the block design has a long duration which could lead to long scanning time and exhausting for the participants. The second limitation of this design is that participants may become familiar with the task, so it becomes predictable and boring. Okay, the second one is called the event-related design. So in an event-related design, tasks are presented in a randomized fashion and the onset between task condition varies. The duration gap between one stimulus and the other is called interstimulus interval or ISI, and it usually ranges between 0.5 and 20 seconds. Because the stimulus is presented at random time points, the design makes it difficult for participants to predict what is coming next and when it will appear. Thus, event-related designs diminish anticipation and boredom. The design will allow researchers to associate brain activity with a discrete stimulus more precisely. This is more possible because brain regions demonstrate different HRF patterns to different types of stimulus. So the researchers could know that, okay, this stimulus would produce this RF pattern and this stimulus would produce this kind of HRF pattern. So if you look at this figure over here, you can see that A, B, and C represent different conditions. Okay, so when we show stimulus A, it will evoke this type of brain activity. When we show stimulus B, it will evoke a different type of brain activity. So by doing this, we would know we would know how the brain responds to a certain type of stimulus. And the unique of event-related design is that there is no specific duration when we will present the stimulus. So in this way, the participant do not know when B will come out or what will come out after A. It can be random. Here is an example of a study using an event-related design. So in this study, the researchers were interested to find out whether former drug addicts will show increased brain activity to drug-related stimuli. So what they did was that they showed the participants several images of drug-related images and several neutral images. As the study employed an event-related design, the images were presented to the participants at different time points. Therefore, the participants could not predict what image will come up next on the screen and when it will appear. This is a clever way to sustain the participants' level of attention and focus during the task. You can see here, the 2 seconds here refers to the presentation of the stimulus. So here ISI refers to the gap or the delay between one stimulus to the other. You can see this one, the gap is slightly longer, and this one, the gap is slightly shorter. So for example, here the researchers show image of drug, and then after a certain period of time, they show a neutral image, and then they show image of drug, and then again an image of drug, and then afterwards, the participant could not predict what image will come up, and so therefore they have to focus on the task given. And then the ISI also differ. Sometimes there is a long gap, sometimes there is a short gap. Also, when using event-related design, there are several things that we need to consider. The first thing is that it requires the researcher to have a good understanding of the type of stimulus and HRF. If not, this may lead to misinterpretation of the results. Second, it requires the researcher to have good data analysis skill. For instance, when analyzing event-related data, in example MATLAB, the researcher must accurately key in the time points in which the stimuli were presented to the participants. Third, a practical rule of thumb is that ISI should be varied, with a minimum of 4 seconds between consecutive stimuli. This is to minimize overlapping HRF. If there is too much overlapping of HRF, the fMRI data may be contaminated. This means that the acquired fMRI signal do not actually present what is actually happening in the brain. Okay, so here we look at the event-related design, and then you can see that the HRF are slightly overlap with one another. So it's important for us to accurately key in the time points when we are doing data processing, so that the HRF represents the task or stimulus more precisely. Okay, an advantage of event-related designs is that it allows stimulus to be presented randomly in one scanning session. It also allows the researcher to estimate the HRF from a single event type and creates less predictable experiments. The limitation, however, are low signal-to-noise ratio and low statistical power. The third design is called the mixed design, which employs a combination of block and event-related designs. The design is commonly used when researchers are interested in understanding the brain's response to sustained and transient activity. Sustained activity refers to the neural activity sustained across trials of a task. An example of sustained activity is the participant's level of attention during a task. On the other hand, transient activity refers to neural activity related to each trial of a task. An example of transient activity is participant responses to accuracy during a task. So here is an image showing the mixed design. There is a block design over here, and there is also a event-related design where the stimulus are presented randomly at random time points. So here is the HRF that would likely appear when using mixed design. You can see a lot of overlapping HRF, and there is a combination of block and as well as event-related in the design. Okay, here is an example of a study using mixed design. The researchers were interested to see how well the participants can get the right answer in the presence of distractors. So in the first block here, there were no distractors, whereas in the second and third blocks, there were distractors. But the number of distractors different between blocks. During the task, the participant had to decide which arrow is presented in the correct position. Here are the two things that will be measured. First was the participant's level of attention, and then the level of attention was considered sustained activity because the participant had to maintain the level of attention throughout the task. And then the participant's decision making, that is whether they make correct or incorrect decision, correspond to the transient activity. So in this mixed design, we could see that the stimuli were presented in blocks, but the order and time of presentation of the stimulus were random. One of the benefits of using mixed design is that it offers the advantages of both block and event-related designs. It is also an effective tool for separating transient trial-related activity from sustained activity in fMRI experiments. In other words, we can differentiate which signal corresponds to attention and which signal corresponds to other cognitive functions like decision making. However, it involves more assumptions. For instance, the attentional level modulates decision making. This can be confusing for researchers who are new to this field. Other than that, it produces poorer HRF estimation. The signal acquired during the sustained activity may not be that strong because it overlaps with other brain signals generated during the task and requires more subjects in order to measure statistically significant and sustained effect. This can be a problem if the researchers do not have adequate funding because MRI scanning can be quite expensive or deal with vulnerable subjects such as drug addicts, because not much drug addicts would like to volunteer in a study. And then the last design is the sparse temporal sampling design. I decided to include this design into this talk because I see studies nowadays are using auditory stimuli. For example, studies have been done to investigate the effects of music or sound on memory performance in healthy and clinical populations. Sparse temporal sampling paradigm is a design where there are long gaps or delays between one MRI scan to the next. The main reason for introducing these long gaps or delays is to allow the auditory stimulus, let's say music or speech, to be delivered to the participants without contamination from the background MRI noise. As we know, the MRI machine makes loud acoustic noise when it is scanning. Therefore, presenting auditory stimuli to the participants during the MRI scan will not be practical. So the best way is to present the auditory stimuli during the silent gap between the MRI scans. The duration of the gap or delay between one MRI scan to another usually ranges from 4 to 12 seconds. The longer the delay, the longer it takes for the MRI scanning session to complete. Okay, here the image is showing that there is a gap. Okay, this one, the gap is 4 seconds. Here you can see the column here represent the scanning, MRI scanning. So here usually there will be a loud noise. And then the shorter the gap, the more rapidly the scannings will be. Okay, this one the gap is 8 seconds. So meaning the first scan will be performed. And then after another 8 seconds, the second scan will be performed. Here the gap is 12 seconds. So meaning the gap between one scan to the second scan is 12 seconds. So between this scan, there is a silent period, quiet period, where usually the researchers would play the auditory stimuli. For example, music or Quran recitation, or as well as speech signal. And then this is also the best time if we want to acquire responses from the participants, because we can clearly hear the response, and as well we can avoid including motion artifact in our data. Here is an example of my own study using sparse sampling design. As we can see, there is a 8-second gap, or delay between one MRI scan to the other. During this silent gap, participants heard a list of words for 4 seconds. Okay, here they hear a list of words. For example, apple, pen, pencil, car. Okay, and then they had to recall the words within the next 4 seconds. So they have to recall back what they heard, but this time in the reverse order of presentation. So doing this, I prevented the auditory stimuli and participant response from being contaminated by the scanner noise. So here there is a scan. The MRI make a loud noise, and then quiet sound. Quiet duration, quiet gap. Okay, and then here the participant hear a list of word 1, 2, 3, 4, and then they have to repeat back 4, 3, 2, 1. Okay, and then after that we scan the brain activity, and then here is the quiet baseline for another 8 seconds. So doing this can take quite some time. So in my case, one condition took about 10 minutes. So I have 4 conditions and total of 40 minutes. Okay, here is the sparse temporal sampling design. Okay, the advantage is that it allows auditory stimuli to be presented without contamination of background scanner noise, and then it also allows participants to respond verbally without inducing motion artifacts. Okay, because we do not scan when the participants are answering the or doing the task. And then sparse temporal sampling, the limitation is only one thing, is that it is long scanning time. So it's not recommended to include too much condition in one experiment. Okay. Next, okay, so the summary. Experimental paradigm is the strategy we use to elicit and acquire brain activity in response to certain stimuli or tasks. And then the experimental paradigm must be designed appropriately to ensure the findings are reliable, robust, and valid. And then experimental design must be determined before carrying out a task-based FMRI experiment. Meaning here to say that before you do an FMRI experiment, you need to design first. And then don't do experiment and then design afterwards. And then before you do your experiment on the actual participant, it is highly recommended that you test your design on a few subjects, and this is what we call pilot scan. And then there is no best experimental design. Okay, we have to remember there is no best experimental design. We cannot say that block design is the best. We cannot say event related is the best, because the best experimental design is the one that answer the study objective. So when you are designing your experiment, the first thing or the important thing that you have to keep in mind is that what you want to achieve, what do you want to measure, and from there you design your own experiment. Okay, here are some of the references that you may want to look at later on. Then, okay, references, okay. Okay, that's it for me. Thank you very much for your attention. Thank you for listening. If you have any question, I'm happy to answer. That's it for me. Thank you.